Hello, everyone. Today we have here the honor to have Professor Scheicher from Uppsala University at Sweden, and he's going to present nano-device for DNA sequencing. I wish you a good talk.
Thank you, Alessandro. Very nice to have this opportunity to talk to you all here. So, as Alessandro said, so the talk is Nano-devices for DNA sequencing. My name is Scheicher, and I work in the Division of Materials Theory at the Department of Physics and Astronomy, and also at the Biophysics Network in Uppsala University in Sweden. And just a very quick introduction for those of you who may be not so sure where Uppsala is. So it's there in Sweden, close to the capital, Stockholm. And the university is actually the oldest university in the Nordic countries. So in the northern countries of Europe, it was founded in 1477. And maybe because it is so old, its campus is a little bit spread out all over the town. So you can see here some picture of the town with the big cathedral here and the castle. And so you see here different departments and different faculties. And we are located here a little bit outside of the center of the city in the so-called Ångström laboratory. And this Ångström laboratory has its name from father and son Ångström, Anders Ångström and Knut Ångström, who were working here, mostly in spectroscopy. And it's continually evolving. So actually this building is around 25 years old now, but they're still building new wings, new parts of it. As you can see, there's some construction here. And so it's continuing to expand. And in the Ångström laboratory, there are different departments. Chemistry is also there, mathematics and physics. And the physics department is subdivided again into different divisions, as you can read here on this slide. And one of these divisions, the one that I'm working in, is materials theory. And materials theory, here we are dealing with different kind of approaches to this topic. So one can subdivide again maybe into 4 different approaches. One of them is computational physics. That's more or less what I'm doing and what I'm going to present. Another one is more mathematical physics, where you use tight binding methods, constructing Hamiltonians for some rather simple model systems that you can then solve maybe analytically or very simply numerically. We have a big chunk of our research on materials for sustainable energy solutions also. And then also some people in our division are actually developing algorithms and codes that can be used for these kind of materials theory investigations that I'm talking about today. So back to the title of the talk. So I want to talk to you today about my research in nano-devices for DNA sequencing. And just to put everybody on the same level, on the same page. So since DNA sequencing is in the title, just talk a little bit about DNA. So DNA, of course, all living organisms on this planet basically have their genetic information encoded in the form of DNA. And it's typically then in the cell, in the nucleus, wound up very tightly in the form of chromosomes. There is this double helix. And this double helix molecule, this DNA molecule, can be studied since about the late 1970s, by using a method called the Sanger sequencing method. So this is if you want to determine the order of the 4 different letters, A, C, G, and T, the abbreviations for these 4 nucleobases. If you want to determine this order, then you have a method available, and that's the Sanger sequencing method. And I will not talk about this method now in detail, But I just want to say that it's well suited if you study relatively short strands of DNA, then it works very well, works very efficiently. But if you want to study longer stretches of DNA that contain many thousands or tens of thousands or even millions of bases in a row, then it's a very rather slow process and quite painstaking, it requires a lot of manual labor, at least in the past. And so it was a very big challenge to apply this Sanger sequencing method to the entire human genome. So if you want to get the order of all 3 billion bases in a human genome, then this is very difficult, very challenging with the Sanger sequencing method. But nonetheless, it was accomplished at the end of the previous century, the end of the previous millennium, 2000. The Human Genome Project was concluded, and scientists in the world for the first time had obtained sort of a blueprint of the human genome, of what distinguishes us from other primates, from other animals, basically, what makes us human from a genetic point of view. But there was a huge price tag involved. So this costs millions, hundreds of millions of dollars and many, many years to complete this project. But as it is with any kind of technology, you've seen this many times for other examples also. And this graph here plots a line here for the Moore's law, which is the famous law for computer technology, how the computers are basically improving or getting cheaper from year to year. Any kind of technology, typically people then try to improve it. And DNA sequencing is no exception. So for DNA sequencing also, there was a lot of improvement after this Human Genome Project was completed. And so you see here that over the course of something like from 2001 to 2007, 2008 roughly, the cost has dropped by about an order of magnitude. And this is reasonable. This is roughly following sort of the trend that we also have in computer technology for Moore's law. But then it dropped even more dramatically. It dropped faster than Moore's law. And you see here, this is a logarithmic scale, so it dropped over many orders of magnitudes in a short time span. This is mainly due to the fact that this Sanger sequencing method was replaced by more efficient methods that parallelize the process, that automatize it, include new approaches basically to sequence DNA. And so now we have 21 years after the completion of the Human Genome Project, we have reached a point where the costs to sequence an entire human genome from scratch, de novo, as they say, is costing no more than around $1,000 and can be accomplished within a matter of less than a day, basically. Even by a single person, this can be done. And so part of this progress is actually due to the involvement of nanotechnology and nano-devices in DNA sequencing. So at first, maybe if you hear about nanotechnology and DNA sequencing, you might think, how does that fit together? Because nanotechnology, I think commonly we associate it to be something that is for the inorganic material world, and DNA sequencing is, of course, something from the biological world, something from the life sciences. So how does these two fit together? Well, actually, they fit very well together if you think about it just from a length scale point of view. So biological molecules, like DNA here, an example, they typically have characteristic features, characteristic lengths that are on the nanometer scale. So for example, for the DNA, these kind of periodic structures there, this minor groove and the major groove in the double helix, they are on the order of 1.2, 2.2 nanometers. And the distances between adjacent bases, so between one base and the next base on a strand, that's even less than a nanometer. That's maybe 1/3 of a nanometer roughly. So just from a purely length scale point of view, if you have a sensing device that operates on the nanometer scale, then it's very well suited to probe biomolecules such as DNA at that same scale, at this nanometer scale. Okay, so what I want to do now is to explain to you through some kind of a schematic and some very simple PowerPoint animations, how can we actually sequence DNA with a nanopore? Just the basic concept so that everybody can understand how one can do this. In practice, there's many technological challenges one has to overcome, but the basic principle I want to present to you now. And it starts with an empty container. So imagine this box here to be some kind of a small container, which is airtight and watertight, basically, and we fill it with water. We fill it with a liquid, and typically just we fill it with water. And in the water, we put some ions at a certain concentration. Can be sodium and chloride, for example, could also be potassium chloride, some positive and negative ions. Now, if we stick then some electrodes inside this container, in practice, this is often silver or silver chloride ion electrodes, and we have an amp meter to measure any kind of current that is flowing from these electrodes to the other electrode, then let's see what happens if we connect it to a voltage source, indicated by this battery here. Okay, let's plug it in. And we see, not very surprisingly, of course, the positive ions, they go to the negative electrode, the negative ions go to the positive electrode. Very simple. And we measure an ionic current. The current might not be completely constant. It's fluctuating a little bit because we have some stochastic going on here. But okay, this is a very simple and maybe a little bit boring experiment. Let's introduce the nanopore to see how we can make it more interesting. So in the schematics that I'm going to show now next, the nanopore is seen from the side. So first I want to show you how it would look like from a top-down view. A nanopore is really just a tiny opening in a membrane. So this membrane here is shown in gray, this gray rectangle here. And now if we have a tiny opening inside there, which is just a few nanometers in diameter, that's what we would call a nanopore. And in reality, it would look something like this. So this is a picture of a real nanopore taken with a transmission electron microscope. And the diameter, the scale on this image is around 15 nanometers. So it's around 15 nanometers wide, this opening in this silicon nitride membrane. In this case, this is silicon nitride. Okay, so in the schematic, I'm showing it from the side. So always remember, when you see this in the schematic now, this is really a circular opening, which is a few nanometers wide, okay? So let's put it in this box. And now something happened to my presentation. Just a moment, please. I'm sorry about this. Let's try to get the presentation back. Just one moment. Okay. Thank you for the message, Alessandro. I hope you can see it now again. Let's see. Okay, I will quickly run to the point where I last was. And it crashed again. Okay. I will try to do it one more time, but more slowly, maybe. Yeah, it was, I think it was back, and then it disappeared again. Let me go to the slide that I just showed last. I think it was this one. I'll try one more time to show to you. Okay, now it should be back. Okay, let's carefully try to continue. This we already seen. Just slowly move to the next slide. Okay, let's hope that it goes okay this time. Okay, seems so far so good. Hopefully nothing bad will happen again. So now we have this nanopore here in this container with the ions in place. And if we connect the voltage source, similar like in this previous experiment, the ions are moving to the opposing electrodes. But now they cannot move anywhere they want anymore. They have to, if they are on one side, they want to move to the other electrode on the other side, they need to move to the nanopore. So think of the nanopore as just being some kind of a resistor, actually, some kind of resistance here. We have a current and we have a voltage that is giving rise to the current. So the nanopore is a resistance that determines the size of the current just according to the basic Ohm's law. So you can already see now maybe the concept of nanopore sequencing. If we change something here with the resistance, but keep the voltage the same, then the current will be affected. So we can use this as some kind of a sensing device, some kind of a detector, if the resistance of the nanopore is somehow affected. Okay, so now here in this schematic, I have introduced some other entity besides the ions, some purple entity, which, as you will see as the animation progresses, it moves inside the nanopore during this experiment, and it blocks part of the ions from moving through the nanopore. So let's see this one. Okay. So if we measure the current, the ionic current, as a function of time, it would look something like this schematically. We get some kind of a constant current, and then it drops to a lower value when this purple entity is inside the nanopore, because this entity has increased the resistance somehow by blocking some of the ions moving through. Okay, now what if we don't just have one type of entity? What if we have different kind, and they have different sizes, or somehow they block the ionic current in different ways, so they all give rise to different ionic currents? Then in that case, we will get different levels of ionic current depending on which entity is inside the nanopore. And if we can calibrate it somehow, then we can read off from the level of the ionic current, we can identify which of these four entities is inside the nanopore, because they block the ionic current by different amounts. So then you can already now make the jump to the DNA sequencing. If we connect these entities by some kind of a linker, some kind of a strand, and so that will be the DNA strand, then the DNA backbone, then we can use this for sequencing. So we can determine the sequence of the entities that occur in this strand by looking at the sequence of ionic current blockades inside this nanopore. And so you probably have all realized now that I'm talking about these entities. These are the four different DNA bases inside DNA: guanine, adenine, cytosine, and thymine. They block the ionic current to different degrees, and thereby one can monitor the ionic current and thereby conclude which sequence of bases we have. Okay, so one question that is, of course, very critical to address maybe is, how can you make in practice these nanopores? Because they have to be very small, and one have to somehow control the process in which we make them, so that you really have nanopores of the proper size and the proper characteristics. Different ways to do it. One of them, as you can see here on the upper left part of the slide, is to actually use the same tool that I've shown you before to visualize them, and that's a transition electron microscope. The electron beam, if it has a high enough energy, if the electrons are moving fast enough, they can knock out the atoms in a thin membrane, and they can actually create a hole, they can drill a hole. And you can use the TEM both to make the hole, and you can also monitor what it looks like, actually. So that is one approach to do. Another approach is actually not shown here on this graph is instead of electrons, you can also use a beam of ions. So for example, helium ions have been used to create also nanopores. So any kind of charged particle, electrons, ions, you can use to bombard a membrane and then create this nanopore. Or you can use something called controlled dielectric breakdown. There you take a membrane and you apply a relatively high voltage across it, and any kind of real material contains certain defects. And typically a defect in a material often comes with a charge. So it has some kind of excess charge, plus or minus. The electric field will lead to some acceleration there, and it will try to move some of the ions there that are in the vicinity of the defect. Typically, this process then creates more defects, and then you get even more acceleration of ions. and so on, until eventually you get this breakdown, until eventually the material breaks down and you really get a channel now where the material allows ions to go through, basically the nanopore that you want to create. And you see this in this experiment. You see this by monitoring the ionic current. In the beginning, when it's not yet broken down, the ionic current will be essentially zero or very, very small. And then when it's broken down, you suddenly see an increase in the ionic current, and then you can quickly, through some electrical circuit, switch it off. And in that way, you can actually, in quite a controlled way, create a nanopore. So this has been done in practice also. And then I will talk a lot about, when I'm coming to the part about our own research in modeling this kind of nanopores, I will talk about two-dimensional materials. And so two-dimensional materials, there's different ways to do it. You can either grow these 2D materials on some kind of a substrate, like shown here on the example of molybdenum disulfide, and then transfer it to some process that is indicated here on a silicon nitride membrane, and then make the nanopore in this 2D material on the membrane. Or you can start with a nanopore in some kind of a, for example, silicon nitride membrane, and then grow through a process called chemical vapor deposition, CVD. You can grow the two-dimensional layer on top of this larger nanopore, this support here basically, and then control the process in some way that you stop before the gap is closing, or you can completely grow a 2D layer here and then you can open it with the help of some electron beam, for example. So these are different ways to create this kind of solid state nanopores. Then how to read out the signal from the different DNA bases. So one of them I've just shown you before in this schematic, this kind of PowerPoint animation, is through the ionic current. But there's also additional ways to detect the DNA bases. And one of them is basically that you allow a current to pass by the nanopore, pass in the vicinity of the nanopore, so that it is affected by the DNA going through the nanopore. So you have effectively two electrical circuits. One is the one I've shown before, where you have these electrodes here that are driving the flow of the ions and are actually also driving the flow of the DNA, because the DNA is typically negatively charged in solution. So if you apply a voltage to these electrodes, the DNA also gets accelerated and moved through the nanopore. And just one comment here, in this picture here, it's a double helix. If you want to sequence DNA, it needs to be a single-stranded DNA. Otherwise, you get a signal from base pairs, and then you don't know whether you have, for example, if it's an AT base pair or a CG base pair, you don't know from which strand maybe the signal is coming. So in reality, if you want to really use it for DNA sequencing, you just run through it a single-stranded DNA, where you took the double-stranded DNA apart, basically. But then this other electrical circuit that I was just talking about before, you have a source here, and you have some kind of like a field effector transistor channel. And the conductance of this channel is affected by the DNA moving through the pore. The charge of the DNA affects modulates the conductance of this channel. So people have experimented with this approach using, for example, silicon nanowires next to the nanopore, or as shown here in this example, they have used graphene nanoribbons. So the electrical current is running through the graphene nanoribbon next to this nanopore, and so it is being affected by the presence of the DNA moving through the nanopore. And then there are also approaches where you actually try to put an electrode that is very sharp, has so almost basically atomically sharp gold electrodes. You try to embed them inside the nanopore, so that when DNA bases are moving through between the electrodes, you get a tunneling current from the electrode to the DNA bases and to the other electrode, and then you measure this tunneling current to try to identify the DNA bases. So, since I will talk later more about 2D materials, I particularly want to talk a little bit more right now about this approach with graphene and graphene nanoribbons. So around 2010, people started to realize that small openings in graphene, so graphene nanopores, could be useful, could be utilized maybe for DNA sequencing. And so this particular experiment shown here on the upper right with this graphene nanoribbon, I want to talk a little bit more about this one. So this was work done by Alexander Radenovic in Switzerland, EPFL in Switzerland. And so here's this schematic that you've seen before. And on the right-hand side here, this is how it actually looks in practice in the laboratory, this microfluidic chambers and all the electronics embedded here in this plexiglass housing. And then if you run the experiment, what you see there is in red, you monitor the ionic current. So just like I've shown you before, you basically get a flat current line, but it's not that flat in reality. In the real experiment, you get some fluctuations. So you get noise basically because it's not always the same number of ions moving through the nanopore in a certain amount of time. But sometimes you get these negative dips. It goes quickly down, it goes back to the noise again. And all of these negative dips are basically events where DNA is quickly moving through the nanopore. Very, very quickly. This is here a time scale of seconds. This is just running for 1 second, the experiment. So if you zoom in, you can actually see a little bit more here, but basically you just see a very big blip, it goes down and goes back up again. Now, if you simultaneously look at the electrical current running through the graphene nanoribbon here, if you monitor that one and plot it here in blue, then you see also some noise, also very noisy, this electrical current. And sometimes you see positive blips, actually. You see an enhancement of the electrical current, and they coincide. These blips, these positive blips, coincide with the negative blips from the ionic current. So it appears so that this is actually really working, that you can really detect through the electrical current, the electronic current in the graphene nanoribbon. You can also detect these events of DNA passing through the graphene nanoribbon. So this was a really nice and groundbreaking work that showed that there are other ways to detect DNA and maybe potentially also sequence DNA than only through the ionic current. You can also use the electrical current. Okay, so that was sort of an introduction a little bit to the background of the field. Now I want to spend some time talking about the research that we are actually doing here at Uppsala University in this field, my research. And so, as I said in the beginning slide, I'm in the division of materials theory. So my research is basically completely done in the computer. So we do computational modeling. And here you see some supercomputer center in Sweden where we're running these calculations. But we're also collaborating with experimentalists. So here in the Ångström laboratory, we have a clean room, we have a microstructural laboratory where people are actually fabricating also these nanopores and doing experiments with these nanopores that we can then compare our theory to. And so in practice, often the way that the research works for materials theorists like me is that there's two possible approaches typically. One of them is that maybe we, or together with the experimentalists, have an idea for what could be a good nanopore setup, what could be a promising nanopore architecture, we say, that could be superior to the existing devices to sequence DNA. And then it would be quite expensive or time-consuming to try to create this in the laboratory, because it's not an easy process to control at such a small level, at the nanometer level, these nanopores in a precise way. But on the other hand, for us computationally, for these kind of simulations we're doing, it is relatively easy. Of course, it's also not that easy, but it's comparatively easier to create such a hypothetical device and then test it in the computer through simulations, how it would perform. And if these simulations are good enough, realistic enough, then they can give us some useful insights. And maybe then, if the results are convincing, we can go to the next step and have the experimentalists fabricate some kind of experimental realization. Or can also be the other way around. Sometimes the experimentalists have already gathered data, they have a certain nanopore setup, but of course, they cannot see what is exactly going on in the nanopore normally. I mean, you can visualize a nanopore with a TEM, but when it's in the liquid environment and DNA is going through, then you cannot have it in the TEM. So they don't actually know. They're kind of blind to what's going on in the nanopore directly. They only have some data. They get data like how much is the current being blocked, how much time does the signal take, and so on. So then they can turn to theorists like me and say, okay, we have this data. Can you explain what's going on there? We don't understand. What is the phenomenon that is occurring in the nanopore? Then through simulations, we can explore the system, of course, in some idealized way, but it's almost like turning a computational microscope to the system and trying to understand the experimental observations through these kind of simulations better. So this is how we typically operate when we are collaborating with experimentalists. And the protocol, sort of the computational protocol for these kind of simulations often follows this schematic that I'm showing here on this slide. Not always, but this is sort of the general protocol that we often follow, that we start from some kind of multidynamic simulation. So we explore the dynamics of the system. We model the system with solvent, with the water, with the ions, with the biomolecules, the nanopore. And because it's a very big system, then we typically have to use some kind of a force field multidynamic simulations. Force field meaning that we have some kind of empirical, simple formulas for the interaction of the atoms with each other. Then from these MD simulations, we can take certain snapshots, as we say. So we take the positions of all the ions, of all the atoms in the system for a certain time, and then we can do density functional theory, DFT, electronic structure calculations to investigate the system in more detail with some kind of a more sophisticated electronic structure method. And then we can go even one step further and sometimes calculate then what is the quantum transport properties, meaning how easy would it be to conduct electrons through this device. So basically the conductivity or even the electrical current we can predict through these kind of methods. And I will explain more this abbreviation, the NEGF, on the next slide. Just want to mention to you the kind of codes that we're sometimes using there, the kind of algorithms. So for multidynamic simulations, often we're using a MD simulation code that is called GROMACS. For the density functional theory calculations, often it's a code that is called Siesta. And for the quantum transport calculations, we have used Smeagol in the past. And here some of these abbreviations are explained. So for the MD simulations, multidynamic simulations, typically we have 1 femtosecond time step. So we calculate the forces and then we advance the system. We calculate the acceleration and the velocity change and the position change for 1 femtosecond time step. And we really have to make it very short, the time steps, so that we get the dynamics right. If we make the time steps too large, then the dynamics does not correspond to reality. If we make it too short, shorter than 1 femtosecond, then computationally it becomes too difficult, too heavy. Then we have to do too many calculations to just get a small amount of time basically simulated. So 1 femtosecond is a good compromise there. And then we try to calculate the dynamics for maybe an iteration of like millions of steps, a sequence of millions of steps. So corresponding to picoseconds or even beyond that, like tens of nanoseconds. Then, as I said before, we can, for some part of the system, we can calculate the electronic structure using density functional theory. And we often use some further approximation method there. It's called the QM/MM method. And this QM/MM method basically means that some part of the system where we are most interested in from a quantum mechanical point of view, we treat using this density functional theory. And the majority of the system, the rest of the system, actually we treat through some kind of empirical force fields and take the effect on the quantum mechanical part only through this kind of molecular mechanics fields, MM, into account. So that makes it feasible to calculate this large system with density functional theory, because the entire system would be too large to treat with this more heavy, computationally more intensive method. And therefore we just concentrate on a subpart to calculate with this DFT. And then finally, this quantum transport, for that we use this NEGF method. It's the non-equilibrium Green's function method. And put in a very simple way, it basically takes the Hamiltonian from this density functional theory calculation, and it takes the electrodes where we inject electrons and take out electrons of the system through some kind of a self-interaction and some coupling matrices. We can basically solve the similar problem that all of us in quantum mechanics have solved for some very simple calculations, just scattering on some kind of a potential. So what is the probability for an electron that comes in with a certain energy, that it makes it from the left to the right side through some scattering potential, basically. So this is more or less what is being done in this non-equilibrium Green's function method for the quantum transport. And we can explore this in the limit of zero bias and the limit of basically no bias being applied, just a zero bias conductance, or we can also apply a bias voltage to the system in the computations and obtain the electrical current. So now I will quickly try to go through some slides basically where I give you some examples from our research. And I don't want to spend too much time talking about each one individually. If you have some questions, then maybe after the talk, I can go into more details if you're interested. But just to give you a flavor, just to give you an idea of what is possible with this computational protocol, what can we investigate and what kind of results can we obtain. Okay, so the first one is one of the oldest papers from my group, and that was from 2011. When just 1 year after, in 2010, this idea with graphene and graphene nanopores came out for DNA sequencing. So we published, we investigated something there which was not actually a graphene nanopore, but rather a graphene nanogap. So we have here basically a sheet, a semi-infinite sheet of graphene on the left side and on the right-hand side, and in between we have a gap that is just large enough that a DNA base comfortably fits inside. So shown here on this example is a guanine base. And so we align it in the plane of this graphene nanogap. And then we explore the transport properties through this approach that I just discussed, through this non-equilibrium Green's function approach, basically treating the left graphene sheet as one electrode and the right graphene sheet as the other electrode, so source and drain. And the base in between and the vacuum gap, that's basically our scattering region. And we're calculating what is the probability for an electron that has a certain energy to make it through to the other side, and then try to get from that basically the current if we apply a certain bias voltage. And this is the result that we obtain from these calculations. So what is shown here in this graph is for the 4 different bases. So this is for the cytosine, so deoxy cytomonophosphate, but doesn't matter. So this is basically cytosine, this is thymine, this is adenine, and this is guanine. And the different colored graphs here, they correspond to this probability that an electron makes it from the left side to the right-hand side for certain rotational angles. So shown here in the legend is the angle by which we have rotated the bases inside the graphene nanogap. So we rotated from 0 degrees in steps of 30 degrees away from this position. And on the x-axis here, this is the energy of the electron relative to the Fermi energy of the graphene electrons, basically. So below the Fermi energy or above the Fermi energy. And basically these peaks, the peaks correspond where you get high transmission, and this is typically so where you have a good coupling to the molecular orbitals of the nucleobases, basically. So the coupling has to be good, the energy has to have the right value, and then you get some large peaks. So this is on a logarithmic scale here, so you really have very large peaks for certain energies. And if you then integrate underneath these curves, that basically the area under the curve can give you an idea for the current that you can obtain there. And so the conclusion from this paper was that, yes, you can basically distinguish the 4 different bases, but there's some overlap in the current. The original paper there on this graphene nanogap, we had the graphene gap terminated with hydrogen atoms. So we had these dangling bonds here saturated with hydrogen atoms, but there are other choices. You can also saturate them with nitrogen, for example. And this is an approach that can actually result in an enhancement of the conductance of the system, and can also actually enhance the distinction between the 4 different DNA bases. So have explored this here, and shown in this graph is basically the comparison between the nitrogen-terminated graphene sheets and the hydrogen-terminated graphene sheets. So always the full line here corresponds to the nitrogen-terminated system that we have focused on here, and the dashed line corresponds to the hydrogenated system. And always we see some kind of an improvement in the transmission. That's understandable, because the frontier orbitals, as we say, from nitrogen, they are reaching out further into the gap. So we basically get a better coupling to the DNA bases. But if we analyze the data further, and I don't have time now to go through all the details, but basically, if we analyze it further, we can also say the distinction, the sensitivity, as it is defined by this formula here, the sensitivity is clearly enhanced for all the nitrogen-terminated graphene sheets compared to the hydrogen-terminated graphene sheets shown here in this lighter color, basically, for each of the 4 bases. So some advantage by functionalizing the edges with nitrogen. And this is what I meant before when I said that we can explore some hypotheses through simulations quite easily, and then this could be translated into some kind of a experimental test. But it's more challenging often in experiment to how do you realize, for example, selective functionalization with different atoms. It's maybe not so easy, but in the computer, since we have full control over where we place the atoms, it is relatively easy to explore this computationally. Another paper that I want to show to you is where we have explored bilayer graphene system for DNA sequencing. So I will show you here, I will walk you through it. So we have a graphene sheet that is connected to the source electrode, and then another graphene sheet that is connected to the drain electrode, but they are not aligned in a gap, but rather they are stacked on top of each other in this kind of A-B stacking that bilayer graphene sheets can have. So again, here's this one graphene sheet representing our source electrode, and we have a pore inside it. And then we have another graphene sheet underneath it, stacked by a distance of maybe just around 3 to 4 angstrom, acting as the drain electrode, and also has a nanopore that is aligned with the top one. And so if the DNA is now moving through these 2 aligned nanopores, what it is doing effectively is it couples the upper layer graphene, the source electrode, to the lower layer graphene, to the drain electrode. So you have some coupling already before, but when the DNA is moving through it, it couples much better. And actually, the distance between the graphene sheets is such that it really perfectly fits basically one DNA base inside there. And so one can analyze then the conductance signal from the source to the drain electrode as a function of time. And the idea is, the hypothesis is here that, I mean, different levels of conductance correspond to different bases. So from the magnitude of the conductance, you can maybe deduce the base level, the base nature. Or you can also, in addition, take into account how long does a base need to propagate through these graphene nanopores. Some of them take longer, some of them take shorter. That depends on the interaction strength. How strongly are they binding to these graphene nanopore edges, basically? So you have 2 sources of information here, the magnitude of the signal and the duration of the signal. And together, then, they can hopefully tell you about the nature of the bases. And indeed, we find that from molecular dynamics simulations and electronic transport calculations here, we find that if we plot the conductance on the horizontal axis and the binding energy of the bases on the vertical axis here, then from the MD simulations, taking different snapshots here, so we have for adenine here these red squares, they all fall within a certain region of conductance and binding energy range. And then guanine falls into this region here. Very little overlap with adenine, actually, so that's good news in terms of distinguishing between the 2 bases. And cytosine, quite interesting, we have a very broad range of conductances here, but they sort of fall into 2 groups, very large conductances and sort of average conductances. And also a group here which is very low conductance, where basically cytosine, being one of the smaller nucleobases, doesn't couple well to the graphene nanopore edges at all. And so we have very low coupling here and basically get almost 0 conductance enhancement. And then thymine, which is kind of overlapping with both adenine and guanine. So in practice, I mean, if this is a realistic prediction, the conclusion would be that this bilayer graphene setup would be quite well suited to distinguish between A and G, but would be difficult to distinguish between T. T could look as an A or a G signal. And cytosine is also difficult because if the signal is in the low conductance case here, then you might miss it. You might basically not see any kind of conductance change there. And yeah, then you cannot be sure whether it's just currently moving from one base to the next and there's no conductance, or it was a cytosine in between. So it would be easy to miss the cytosine signal. Another system, I think the second last system that I want to show to you now here today is one that we came up with, where you have a graphene nanorode, shown here in gray is the graphene basically, embedded in hexagonal boron nitride, shown in this purple and blue kind of atomic model here. So to either side you have hexagonal boron nitride, and the nice thing or the interesting thing about hexagonal boron nitride is that it is insulating. It does not conduct electricity very well, and graphene does. So basically the current, the electrical current coming from some left lead here, some left electrode going to the right electrode, is constricted first of all to this graphene nanorode. And in addition, if you drill some kind of a nanopore inside here, and you spare out one layer of carbon atoms on this side of the edge here, then the atomic wire that is created here is conducting the electricity. So it's very sensitive now. We have basically, again, this kind of field effector transistor setup for an atomic wire. So anything moving through this nanopore here, if it is charged, if it has positive and negative charges, might affect the conductance of this atomic wire. And then you can read that out by a change in the electrical current or the electrical conductance. So we've done this test, we've done this simulation in the computer. So we've put the 4 different bases, adenine, guanine, cytosine, and thymine inside this nanopore, and then calculated a transmission curve, compare it to the empty pore, shown this black dashed line here basically. And in some cases we see an enhancement of the conductance, in some cases we see a drop in the conductance. And we can explain this if we look at the polarity of the side groups of the DNA bases that are facing this atomic wire. So the different bases have different kind of side groups. Some of them are negatively charged, some of them are positively charged, and that affects the conductance in a characteristic way. And so we can distinguish for certain gate voltages here between the 4 different bases quite well with this setup. And this setup we have actually explored for many kind of different cases, also for other molecules and so on. It's a very nice setup that is, has very high potential for sensing biomolecules and distinguishing between them. And the last paper, one of the most recent works that we've done in the field of like 2D materials for DNA sequencing, this is with something called MXene materials. So they all have some characteristic chemical formula here. This one is titanium 2 carbon, and then it's functionalized here with hydroxyl groups. So basically, again, similar setup. We have a source electrode, a drain electrode, and also a gate electrode. I didn't talk so much about this before, but basically the role of the gate electrode is to shift the Fermi level or the chemical potential of the entire 2D material to different points in the energy, and thereby can make it more sensitive, the pore. So again, we have this nanopore, we put the different bases inside here. And this was also involving MD simulations. And so here we see again the conductance, the transmission as a function of energy of the electrons injected here, and compared to the empty pore. And if we do some statistics here, if we look at make some histograms of how many times we get certain kind of conductance values for the different bases, then we can analyze here that for different gate voltages, we do see some distinction between the 4 different bases again. And we can also analyze the binding energy. We can analyze how strongly the bases bind to the nucleobases, which would then again result in different durations of the signal, just like I've shown before for this bilayer graphene system. Okay, I think because the time is maybe getting a little bit shorter. Just in the last part of the talk, I want to talk a little bit about the actual real-life applications of DNA sequencing with nanopores, because those exist, and I didn't talk so much about it so far yet. So I put this question here on the screen: is it actually possible to sequence DNA with nanopores? And the answer is yes, but there's a but. So first, the yes. So the company which has commercialized nanopores is from the UK. And so around 2012, 2013, they started to come out with a product which they called the MXene. Maybe explain more why it's called this way. And now they have a whole line of products for DNA sequencing, which utilizes nanopores actually. And the way they do it is actually a little bit different from what I've talked so far about. So I have focused completely on this solid state nanopores in silicon nitride or in 2D materials, but actually they are using biological nanopores. So these biological nanopores are basically quite complicated protein complexes that exist in nature. So there are certain kind of organisms that generate these kind of nanopores, and you can utilize them here for this purpose. So you have some membrane, which in this case is a lipid bilayer membrane, and these nanopores, which are shown very schematically here, embed themselves in these membranes. And they have some kind of a funnel here, basically, and they have a small constriction here, which is then the sensing region. So it's cut open so that you can see inside here. And this is an MSPA nanopore, for example. This is an alpha hemolysin nanopore. Doesn't matter so much. What is interesting is that they have different sensing regions. So some of them are smaller, some of them have larger sensing regions. For the purpose of DNA sequencing, you want to have a small sensing region because then the signal just comes from one base or a few bases, basically. And the reason why this company, this Oxford Nanopore Technologies, who has been successful in in transforming this nanopore sequencing idea into a commercial product, why they have been successful is because actually they are able to slow down the motion of the DNA. This is a big problem for solid-state nanopores, and in principle it is still unsolved. The DNA can be translocated through a nanopore, but it's moving through very, very fast. And too fast to basically read out a signal to distinguish between the 4 different DNA bases. But for biological pores, what you can do is you can introduce in the solution also an enzyme. And this enzyme in particular is called here Phi29 DNA polymerase. Doesn't matter so much the details, but what it basically does is it grabs on to a double-stranded DNA and it takes the double-stranded DNA apart into the 2 single strands, and then it moves these single strand, one of the single strands, one base at a time, very slowly through. So it holds on very tight to this DNA double helix, and it allows the single-strand DNA only to move very slowly through the nanopore. And so it's this kind of ratcheting motion. So like moves and then moves again and moves again. So it moves stepwise, basically. So you have a lot of time when the DNA is basically not moving to get ionic current passing past the DNA bases, and thereby you get a very stable signal. So that's the secret to their success, basically, why this Oxford Nanopore Technologies was able to build a device that can actually use nanopores to sequence DNA. And so this is how it looks like. So the most successful of their devices is this MXene, as I said before. It's not much larger than like a bigger USB stick, basically, and has everything here. When you open it up, basically, there's some part where you deposit a drop of the solution containing DNA, which you maybe had to purify before a little bit and clean it so that it doesn't clog the pores. But basically, you deposit here a drop of the liquid containing DNA, and then it's connected through a cable to a computer running the software from the company. And the cost is around $1,000 for this MXene. And you can use it several times actually to sequence DNA. And it has been used over the years. Now these studies are a little bit older, to study, for example, the evolution and the mutation of viruses, or of RNA from viruses. The Ebola virus, for example, in Guinea, that was 2014. It was studied there how these mutations occur, and transmission history and so on. And also in Brazil, actually, it was used during the Zika virus outbreak. And so the advantage of this MinION sequencer is basically that one can very easily conduct this kind of studies sort of in the field. One does not have to collect samples and send it in to some central laboratory, and that takes a long time to analyze and so on, but one can do it sort of there on the spot and save time. Therefore, it can save resources and help maybe the health, the public health authorities to make decisions based on the results very, very quickly. Okay, I think the time is getting a little bit short, but I just very briefly maybe want to talk about some very exciting new application of nanopores, but I will try to go through it quickly so that we don't spend too much time. And so DNA sequencing, sort of from a technological point of view, is a solved problem. So there exists a company that markets these nanopore sequencing devices. What is the next frontier? What is the next application target for nanopores? And very much in the field, what is being discussed now for the past few years is that we could also use it to sequence proteins, actually. Okay, so single molecule protein sequencing was actually was nice to see it highlighted earlier this year, in the beginning of the year 2023. Nature, they always have this tradition somehow. They select like 7 technologies. I don't know why it's 7, but they always select 7 technologies to watch for in the coming year. And the beginning of 2023, one of the 7 technologies, and actually the first one that they mentioned in this article, was single molecule protein sequencing, and nanopores are also mentioned in there. So it's a very hot topic, I would say, very things where progress is being made currently very rapidly. But just to give you an idea of the challenge here, so we have for DNA, we have only 4 different bases to distinguish. For protein sequencing, proteins are made up of 21 different amino acids. So that's already a challenge, that you basically get a signal from 21 different entities that you need to distinguish. So that's a big challenge. There's more challenges in the sense that DNA behaves very nicely in solution. If you have electrodes, they move towards one of the electrodes because DNA is negatively charged. Proteins, they don't do you this favor because they come with amino acids that some of them are negatively charged, some of them are positively charged, some are neutral and just have some polar side groups basically, and so on. So just the question of how to even translocate, how to move a protein chain through a nanopore is already it's a good question, basically, how to do this in practice. So people are discussing sort of 3 different kind of strategies how nanopore-based protein sequencing could work. And so the first strategy here, so number 1 here, is that, okay, we unfold the protein. So we take this 3-dimensional structure of the protein apart. We just have a long chain now of amino acids. And shown here is the abbreviations of these amino acids. So they also get letters, each one corresponding to one type of amino acid. And as they move through the nanopore and we monitor maybe the ionic current, we get some signal as a function of time. And then through some signal processing, but this is of course not, this is the key problem here, how to actually do the signal processing, we get the protein sequencing out. Okay, I'll talk a little bit more about this strategy number 1. Number 2 is actually people very cleverly came up with the realization that you don't have to know the order of all of the amino acids in a sequence in order to identify which protein is actually going through a nanopore. If you can only identify 3 amino acids, actually, just C, K, and M, the order of those uniquely identifies the protein in, I don't know, something like 98% of the cases. So if you go to some database, the protein database, and you search for which protein has this order of K, M, C, and so on, you can find basically just 1 or very rarely 2 proteins that have this exact same sequence. So you don't need to know the, the nature of the other amino acids in between, or even how many there are in between. And how can you distinguish between those 3? People are talking about using some kind of fluorescent labeling. So you label them differently with different fluorescents that attach to them. And then as they go through a nanopore, exciting there with a laser, you basically get fluorescent signals that are different for these 4 different bases. And maybe you can couple it also to the current signal And then through some kind of, again, signal processing, protein classifier called here, you can say, okay, it's such and such protein. You go to the protein database and then you have identified the protein. So that's strategy number 2. And the last, strategy number 3, not utilized, I mean, not really utilizable maybe for sequencing, but if you just have maybe a few proteins that you want to distinguish, maybe it's enough to just run them in their folded form through the nanopore. And then you also get some kind of a signal. And maybe, again, through some magical signal processing, you can identify some protein properties. And so this strategy number 3, we actually, about 2 years ago, we investigated a little bit here. No, 2 years ago, we did the research, and last year the paper came out. So we looked at 2 different mini proteins, which have some names here that you don't need to remember, but they were quite small, and they fold up in structures that look quite similar in terms of a 3-dimensional shape and in terms of the electrostatics of the surface. So this is this one mini protein, rotate around a little bit. This is the other mini protein. So yeah, it looks quite similar, has a similar kind of volume, similar kind of shape, basically. And how could you distinguish between these 2 using the ionic current in a silicon nitride membrane? That's the question that we wanted to address here through some computational study, through some simulations. So we constructed a nanopore in silicon nitride here, which has a channel, a cylindrical channel of around 3.6 nanometers or 36 angstrom. This is the top view. This is how it looks like when you cut it open. And then we put those 2 different mini proteins inside, starting in different configurations, different orientations, and measure the ionic current in the computer, meaning just basically counting how many ions are moving through the nanopore as a function of time. So if we measure the ionic current in the computer, it really just means basically calculating where are the ions, the different ions, there are n different ions with different charges, plus and minus. What is their z-axis position at a certain time t and a certain time delta t later. And typically we do not get too much noise here. We take this time step 1 picosecond. Otherwise, I mean, there's a lot of erratic movement of the ions, and then we would get a very big noise. And to get a current out of it, then we have to divide through the length of this nanopore channel. And the raw data for one of the mini proteins looks like this, so very noisy. If we filter it, so some kind of a signal filtering processing, okay, then we get something which is a little bit more well-behaved. So we do this for like around 40 nanoseconds or so for the 2 different mini proteins. We can do it at different bias voltages, and then basically what we get here is like different distributions of the ionic current. But they overlap quite a lot. So you see for these 2 different mini proteins, we can basically not distinguish between them just on the ionic current alone. But then I talked already about this before in my presentation a few times. Besides the height of the signal, we can also talk about the duration of the signal, how that affects how that's affected by the different mini proteins. So the simulations, we cannot really run the simulations long enough to statistically reliably observe the translocation of many mini proteins. We can run it for like 40 nanoseconds or so, but then in some cases the mini protein will not move very far during this time. So we had to come up with something a little bit more clever. And so what we came up with here is that, okay, all these proteins, of course, they're made of amino acids, and the amino acids have different kind of side groups. So if these side groups are interacting with the silicon nitride wall of the nanopore in different ways, then maybe we can use that interaction energy to say something about the binding energy and therefore the translocation time. So we did this in this project here. So we investigated how all the different amino acids, all the 21 different amino acids interact with silicon nitride through some kind of density functional theory calculations. So it follows this kind of like a Morse potential here. And we obtained then the binding energy for all of them. But of course, not all amino acids in this different mini proteins are equally distributed on the surface. Some of them are deep inside the protein. And so what we've done is construct some kind of a map, basically, which says how likely is it to find an amino acid close to the surface, basically. And taking this distribution into account, together with the energies, the interaction energies calculated from density functional theory, we obtained then finally some interaction energy for the two different mini proteins. Here for this one mini protein, here for the other. And yeah, so on average, one of them interacts with around 5.57 eV with the nanopore wall, and the other with 7 eV, and quite some large standard deviation. But still, the binding energy goes into the translocation time, how fast the mini protein translocates. So actually, this results in translocation times that are on average orders of magnitudes different from each other. So here we have like around 394 nanoseconds, so 0.394 microseconds. And here we have only like 31 nanoseconds, much faster, in this case, the translocation process. So this should be distinguishable. I mean, the duration of the signal should be able to distinguish because it's orders of magnitudes faster in one case compared to the other case. And actually spread out quite a bit. So it spreads out over one order of magnitude, but the overlap is almost non-existing here in the translocation times. Okay, yeah, I wanted to talk about strategy number 1 a little bit, but this is not my own work. This is work done by some other group, basically. And so I just very, very quickly go through it. This is just a very nice paper in Science from 2 years ago. Experimentally, this is where they can resolve single amino acids using nanopores. And the approach is basically that they link the peptide, the protein that they want to study, to some DNA strand, and use this DNA strand, pull that one through the nanopore, and then it pulls with it the peptide that they want to analyze. And it involves, again, some kind of an enzyme. Instead of polymerase, this is called helicase, but it functions similarly, but it attaches to a single-stranded DNA, and it moves the single-stranded DNA slowly out of the pore, and thereby also pulls out the peptide through the nanopore, and then you can get some ionic current signal from the peptide, actually. So here's a different kind of schematic that shows this more nicely. Basically, it's like almost like this kind of a ratchet. Basically, it moves the DNA upwards, pulls it out of the pore, and then when it reaches the peptide, this helicase, it doesn't know what to do with peptides. It's not designed to interact with peptides, so it just falls off. But there's another helicase already attached to the DNA higher up on the strand. So then the whole thing is like sucked into the nanopore again, and then it's being pulled out again by the helicase. This process repeats and can basically repeat as many times as you want. And so you read the amino acids in the peptides many, many times. If you make an error in one reading, then in the next reading, it's probably going to be corrected. So it's a process that after a certain number of reading the peptides, the identification accuracy actually very quickly converges asymptotically to 100%, basically. So if you just read maybe like 20 or 30 times the same peptide chain, you have almost complete certainty that you read it actually correctly. So this is a very nice approach. It's nothing anywhere near something that you can commercialize yet, but it's a nice proof of principle that with this approach, you can actually identify different amino acids, at least a few of them. This was not all of the amino acids, I think, with this nanopore approach. Okay, I think it's really time to wrap up. So I conclude with 3 brief summaries here. The first one for this nano-devices, because this was what the talk was sort of about. So for nano-devices, I would say that these 2D materials, they're very well suited to study biomolecules because they are atomically sharp, so they can really couple to individual parts of the DNA or proteins very well. The kind of investigations that we're doing, this atomistic modeling, can help to investigate new kind of designs in the computer and can thereby sort of simulate and in the computer investigate architectures, nano-architectures that are difficult or challenging to construct in the laboratory. There's many challenges that I didn't talk so much in detail about that still need to be addressed. For example, how we actually make the pore without destroying some of the nice properties of the 2D material. I didn't talk at all about that you need to also include some protective layers. For example, if you have graphene, if you just put it barely into the the liquid environment with the DNA. DNA will stick to it, so you need something that prevents DNA from sticking to it, and so on. But yeah, because there's so much variety in creating 2D materials, you can have them in, you can take different combinations like hexagonal boron nitride and graphene, and there's also so many different kind of 2D materials, new ones being discovered all the time, that it could be an advantage compared to like solid state nanopores or even biological nanopores using this 2D nanopores. Then summary for the DNA sequencing of nanopores, as I've tried to show you. So from a commercial point of view, it's now reached the $1,000 per genome threshold. So it's quite cheap. You can sequence with very long base read lengths, so millions of bases in a row. It's very good if you want to assemble a genome from scratch, de novo, basically. It's a single molecule technique, so you don't need to, in principle, amplify the DNA. You don't need to make copies of the DNA before, so it's fast. And also it can detect modified bases. I didn't talk about this at all, but this epigenetic changes in DNA bases, if you, for example, methylated cytosine, it can detect them directly through a change in the ionic current. You don't need any chemical manipulation. And the devices are small, so you've seen this mini-ion device is very small, it's portable, you can use it in the field also. For protein studies, yeah, nanopores again, they offer the possibility to detect and characterize proteins and protein chains at the single molecule level, which is particularly important for proteins because they're the life scientists, the biologists, they don't have a method to easily make copies of proteins. For DNA, it's easy. You have this polymerase chain reaction, PCR. But for proteins, you cannot easily make copies of them. You have to work with the material that nature or some kind of sampling method basically gives you. And you can use the nanopores to obtain information on a wide range of protein properties listed here, concentration, shape, size, etc. And you can study also very fundamental processes in biochemistry. So, for example, the unfolding of proteins and protein translocation and so on. So yeah, it's lots of very interesting ways to use nanopores for the study of protein biochemistry also. So I want to wrap up with an outlook to the future. So it's maybe interesting if you would think like some kind of a time travel, like 20 years ago, at the turn of the century, I've shown you before, there was this conclusion of this human genome project, and it costs so many, like hundreds of millions of dollars, and so many people were working on it. If you would have said to people at that time, oh, in the year 2013 already, like so just like 13 years later, there will be a device that can do all of these things for $1,000 and it can be operated by a single person. It takes less than a day to get the sequence of the genome of an individual human. I think most people would not have believed it. I mean, maybe some people who are visionaries would have said, yes, this will happen, but I think most people would have said, no, this is science fiction. This is so far in the future. We cannot imagine this yet. So we have to see how fast technology progresses. It's an exponential process, basically building on the previous discoveries, and it only goes faster. So now, right now, for protein sequencing, we are at the same stage again, basically. A lot of people are seeing a lot of challenges, and we don't know yet exactly how to solve all of these challenges. But I would really bet, I'm very sure, that in a similar amount of time, like it was possible for DNA sequencing, or maybe even a shorter time, it will be possible to sequence proteins with nanopores. So we can really use this technology also to study not just the genome, but also the proteome. And that's incredibly useful for people who are working in life sciences. Okay, now it's really time to stop. The last thing I just need to do, most important slide of the whole talk, is to thank all of the people who were involved in this project and who really, I mean, made it happen, basically. So it's a big collaboration with many fantastic people from Brazil, also from other countries, from Thailand, from China, from the US, from India, from Germany, and so on. So I'm really grateful to all of these people, and also funding that we have received from various organizations. I also put CAPES there because CAPES allowed me to visit Brazil recently, to stay there for 2 weeks. And yeah, the last thing that I have to say is just thank you, obrigado, for your attention. Thank you.
Thank you so much for your talk. And we do have a lot of questions here.
Great.
I think well, the first one is from Professor Valdemir. He's a professor here at the university. He says, very interesting seminar, and he thanks you. And he asks, could you comment a little on semi-empirical methods, for example, tight binding, and wouldn't these be interesting for a system with such a large number of atoms?
Yeah, so in the literature, there are indeed quite a few studies that have used these tight binding methods. And from my perspective, one of the problems there is what parameters do you basically put into your model for the hopping, for example? So you need to somehow what you want to have some kind of a coupling again between some electrodes that are in the nanopore, or some coupling of some conductor that is running next to the nanopore to the DNA bases, basically, where electrons can jump or somehow influence the conductance of these atomic wires. So I think this is the challenge. I mean, of course, you can construct these tight binding models, but in order to make realistic predictions, somehow you have to still maybe link it to some kind of ab initio or first principles calculations to set the parameters to the right value. But if one succeeds in that, then I think tight binding models can be very useful, because you can, I mean, they're so much faster than the methods that I talked about here, and they can really be also useful in this kind of system to make predictions. It's a very good comment here. I think you're muted, Alessandro. I cannot hear you. Sorry.
Yeah, sorry. Next, we have a question from Marlon. He's a friend of mine. I think he's a master's student. He will be in PhD soon.
Okay, good.
He asks, can we use nano-device to identify which medications were bound to DNA during a treatment, where they bound most preferably, to compare them with other medications?
I see. Yeah. It's a very good question. I'm not sure if I'm qualified to answer it completely. Because I'm a physicist and my background is not exactly in biochemistry, but I have seen studies where people have trapped biomolecules, maybe including DNA, in the nanopore. So in these studies, you're trying to actually not translocate the DNA through the nanopore, but it's in the nanopore and cannot move any further, for example, because it is bound to something that is too large to fit through the nanopore. And then you can sometimes then you can study through the evolution of the ionic current as a function of time. You can study that the molecules undergo different kind of configurational changes, and you can interpret then the change in the ionic current as a function of time with respect to these changes in the configurations. So that's where it's very important that I talked about before, to have this kind of simulations, because there you can then interpret the experimental signal and make sense of it sort of from an atomistic point of view, and see really what is happening in detail. And so some of these, these studies could be used to maybe also investigate on which part of the DNA strand maybe certain molecules that are like drugs for medication could bind. So unfortunately, I cannot say any more details than that, but in principle, it is definitely possible to use that. And maybe these studies have already been done, but I'm not fully aware of. The literature is very, very big now. It's grown a lot over the last 10 to 20 years, but it's definitely possible. It's a good point, yeah.
Great. We have another one from Professor Valdemir.
Mm-hmm.
He says, you mentioned water in your simulations. Do you really need to include water explicitly, or would it be enough to include in the form of a mean field, PCM or similar?
Mm-hmm. Right. We investigate exactly this question quite recently. I didn't have time, unfortunately, to include it now in this presentation here. But Yes and no. So I think in some cases you can really make predictions where you just take some kind of an average effect of the water into account. If you're not interested in the details, that might be fine. But we found actually that in two cases the water is very important to take into account. First of all, the dynamics of the DNA molecule. How it interacts with the water, the fluctuations, the geometrical fluctuations, and so on. It's really important to take the water through some kind of a force field approach explicitly into account. If you just do it through a mean field approach, my understanding is, and my intuition tells me that the fluctuations would not necessarily be the same. But we've seen one case where it's really important even to take the water molecules quantum mechanically into account, and that is this system where you have, for example, this nanogap that I've shown before. So where you have like one electrode here and another electrode there, and a biomolecule running through in between. And there we saw that if you don't treat these water molecules as part of the quantum mechanical system, you get different results actually than if you actually include them in your quantum mechanical, in this density functional theory calculations. So we really get a coupling of the orbitals from the electrodes to the water molecules, to the biomolecule, to the water molecule, and to the electrodes again. So the water is really playing an important role. And if you want to make like quantitative predictions, because this is always the goal, I mean, we always try to make the models as realistic as possible, but of course, keeping them computationally manageable. If we want to make as good quantitative predictions as possible, then we have to take the water explicitly into account in the model.
Okay. Now we have a question from Benny. Benny asks, is there any reason for the specific use of graphene? Maybe something related to the structural characteristics of this material?
So graphene was chosen as the first of the 2D materials, mainly because in 2010 there were not that many 2D materials yet. So this was like shortly after the first time that graphene was sort of extracted as an individual 2D material, but it has certain nice properties. I mean, one thing that all 2D materials, of course, have in common is that they are at the edges, they are atomically sharp. So basically, one can form a coupling to the biomolecule. Basically, you cannot make something that is thinner than one atomic layer in principle. But from an electronic structure point of view, it is also nice that the graphene basically has this kind of a semi-metallic nature. So if you apply a small bias voltage, basically you get a small current here, and then it's good in some sense that the current is not too large, because you can observe modulations then better in the electrical current than if the current would be very large. But you need something, of course, at least for this electrical conductance case, you need something that is conducting. So it wouldn't work if you would have some kind of a material that is an insulator or a semiconductor with a large gap. So it needs something that conducts the electrons and sort of conducts the proper amount of electrons. So this, together with this atomic sharpness, that's why the graphene was kind of special, but it's not so special anymore. So now there's more and more other 2D materials also being explored for this sensing.
Great. He also asks that once the DNA sequencing is applied to the research, it is possible to extend the results to the treatment of some diseases. Yeah.
So unfortunately, I didn't have time to talk about that in detail, but the big motivation for this is, I mean, DNA sequencing now is really something that can be done in a clinical setting for a relatively low price tag, as we saw. I mean, it costs in principle just $1,000 and less than 1 day to sequence the DNA of a human genome, 3 billion bases. So one approach for clinical settings where it's already being used, as far as I know, is to actually extract a small sample from a tumor, so if one has cancerous growth, and determine the sequence of the tumor where you have some mutations, and then through this analysis, through the mutations, one can then decide what treatment maybe works best for this particular cancer. So it's really now starting to become this kind of, they talk about personalized medicine or precision medicine, where you don't just have one approach for all patients, but you basically tailor the approach, the treatment to either the genome of the patient or maybe the sequence of some kind of mutated tumor sample and so on. So that is one approach, for example. But I think it will come more and more. I mean, I think we are very close to that in the future, in the near future, maybe for all kind of diseases and for all kind of treatments, the human genome can be taken into account, and maybe one can get information from that analysis why what's the origin of a certain disease. Maybe it has some connection to the genome of that person, of that patient, and also what kind of treatment, what medicine might work best, because that is the reason why some people get side effects and other people don't experience side effects. It can often be linked maybe to the genome of the person. And then if we go one step further, if I just connect it to what I talked last in my presentation, this proteome studies. So this is sort of, that would be even a further step, because the genome basically contains the instructions for making proteins in the body. But of course, not all possible proteins that can be made according to the genome are also being made, are expressed, as they say. So if we could use this nanopore approach to study proteins, one can imagine an approach where you take all the proteins from a single cell in a patient that you want to analyze, and then use some approach like nanopore's protein sequencing to count how many different proteins you have expressed in a certain cell. And that can also give a lot of information. Basically, you can say what is sort of a normal or a healthy level of different proteins being expressed. And maybe if you take a sample from a person, maybe it has very abnormal levels for certain proteins, and then that could be a sign that there's a certain disease maybe ongoing, or it can maybe explain why the patient has certain kind of symptoms and so on. So it contains, it's a very precise tool for diagnostics and also for deciding on treatment. And I think we are really now in a time where it's getting very close to being reality in the clinical setting, that this can actually be used to help patients to get the best optimal treatment that is possible.
Yeah. That's deep. It leaves us kind of speechless. Well, the next question is actually a curiosity. I would like to know, is there any use of artificial intelligence to recognize the amino acids?
Yeah, for amino acids, I'm not so sure yet, because this is still a very new topic for the protein sensing. But for DNA sequencing, there have been papers published where basically the ionic current that you get, for example, in a nanopores as a function of time, it oscillates a lot. You have a lot of noise. And to make sense of the signal and noise, I mean, of course, signal processing is something that exists for a long time, and people have been using different approaches. But now with this explosion of machine learning and artificial intelligence, people have also applied that successfully to get sort of extract the most useful data out of the noise, so that you basically can really utilize all the information that's in the signal, which is maybe missed with some more traditional approaches. So the answer is yes, this is something that is actively being used and also being investigated in articles being published, to use machine learning to optimize the signal processing there for DNA sequencing. And I think it will also come for protein sequencing, but because it's a new field, I haven't seen so much yet done in that respect.
Okay. I think that's all. People, thank you for the talk, and yeah. So thank you, everyone, and you can say anything you want. If you want
No?
to share your contacts,
Yeah, exactly. So I put there on the slide. If anybody has some follow-up questions, so you can see my email address there, and also my Twitter handle basically is where I sometimes post some things related to papers that we publish and so on. So yeah, I'm welcome to contact me. I'm always glad to communicate and discuss about science, especially if it's related to nanopores. But I'm a scientist. We are all studying science, so communication is always good. It's always good to communicate our ideas.
Okay, so thank you everyone, and we'll see you later.
Yeah, thank you very much again. I'll disconnect now. Thanks, Alessandro.
Okay.
Okay.
Bye.
Bye. Bye.